Every great research project begins with a question. For us, it was understanding the impact of invasive signal crayfish in Croatian freshwaters. What are their food preferences? What is their niche in the trophic food chain? And how do they compare to native crayfish species? With these questions in mind, we started the journey to the wild Croatian rivers. Before stepping into the field, I spent months designing the study. Field trips were planned, transportation arranged, equipment sourced, and permissions secured. Collaboration was key, as I had to gather a small team of experts to help me bringing this project to life. In the summer of 2023, the real work finally began. We spent 3 weeks in the field, placing around 20 baited traps each night to capture our freshwater subjects, crayfish. During the day, we sampled potential food sources like insect larvae or snails, various plants, and decomposed organic matter, all crucial to the diet of this omnivorous species. But fieldwork isn't without its challenges. Keeping samples cool was one of them. Luckily, a friendly pizzeria in the middle of nowhere at one of our spots, the Drava River, lent us their freezer so our samples could stay cool during fieldwork. At another river in central Croatia, the Korana, I received help from the local environmental agency, who provided valuable assistance in locating the best sampling spots and arranging cold storage as well. Of course, nature had other plans. Low crayfish numbers, high water levels, and stolen traps tested our patience. The terrain and way too many mosquitoes were a major challenge for us sometimes. Additionally, at the Korana River, we found hardly any native species at all. Despite the hurdles, we pressed on, refining our research questions to focus only on the invasive signal crayfish and see if this species shows some differences between the rivers and different invasion stages. When sampling was finally done, the over 200 stored samples from each location were brought to the University of Zagreb to be stored at -25°C to ensure everything stayed fresh until the next steps. Then, in end of autumn, it was time to analyze. All samples were measured, weighed, invertebrates were identified as precisely as possible to their functional groups, and everything was freeze-dried and powdered, preparing us for the next phase. Fast forward to winter, and I'm off to Estonia. From the warm, sunlit rivers of Croatia to Estonia's snowy north. At the University of Life Sciences, I trained for 2 weeks in a method called stable isotope analysis. Then it was time for me to hit the lab in Tartu University and prepare each sample precisely for the mass spectrometer. This is where the magic happens. By analyzing carbon and nitrogen isotope ratios, it is a neat way to get insights into the crayfish's diet and their place in the food web. With raw data in hand, we dove into complex analyses and transformed the numbers into compelling graphs and results. Our graphs tell a story of invasion, adaptation, and the shifting dynamics in these ecosystems. Now, the next steps are underway, crafting a manuscript for publication and sharing our findings at ecological conferences, sparking discussions and fostering research exchange within the scientific community. And that's a glimpse of what this project was all about, helping us understand invasive species like the signal crayfish and finding ways to protect our precious ecosystems. Science is an adventure, one that pushes not only the boundaries of science, but mostly our own, and brings us closer to preserving our precious ecosystems.